Okay, so good morning, everyone. Thanks for attending bright and early on the first day of JP Morgan. And I appreciate the organizers for giving us this platform. When Betsy asked the question, what topic would you like to do? I think the whole world and their moms were going after oncology. I said, we'll do something new. And that's how we landed on, I mean, we have been doing some kind of landscaping exercise around what might be the quote-unquote orthogonal areas, not quite in focus, but still very important unmet medical needs. And we landed on autoimmune disease and inflammatory diseases. So then we went about selecting and sort of cherry-picking a group of companies and their CEOs, CSOs, to really give you a flavor for a spectrum of approaches that have been sort of bubbling up to the top over the last 3, 4, 5 years, but just beginning to get some headlights on them. And this was a very deliberate selection of companies and their execs. We unfortunately don't have Asthika today because he got mired in some kind of compliance issue because he launched coverage today. And he would have been our Wall Street analyst. So I'll try to play the role. It's difficult to be Asthika, though we either don't have hair, but we almost look the same. But it's difficult to play the role. But nonetheless, we have 4 great companies here. And without further ado, maybe we can do a quick round of introductions before we jump into sort of the framework of the discussion.
Hi, my name is John Honecker. I'm an oncologist leading Anokion. We're a biotech company that is focused on treating autoimmune diseases by re-educating the immune system. Our platform is an antigen-specific immune tolerance platform.
Hi, I'm Gwendolyn Binder. I'm the Executive Vice President of Cabaletta Bio. We are an engineered T-cell therapy company focused in the treatment of B-cell mediated autoimmune diseases located in Philadelphia, Pennsylvania.
Hi, I'm Ben Yao, CEO of Viela Bio. We are focused on autoimmune inflammatory conditions. We are using this shared pathway approach. So rather than you going one disease at a time, we're targeting critical pathways that are shared by multiple different types of autoimmune conditions or subsets of the disease, with 5 products in development, 3 in clinical stage.
Good morning. My name is Ruben, the CEO of Polyneuron Pharmaceuticals, a Swiss startup also in the autoimmune field. We work with a platform which enables targeted depletion of autoantibodies.
Perfect. So as you can see, this is a good cross-sectional representation, so to say. Why do I say that? Because we have 2 public companies represented by Gwen and Bing, and 2 private companies in different stages of development. We have different modalities here and fundamentally different approaches that they are taking. So why autoimmune disease? The rationale was, if you go back 20 years, oncogene and the directed, mutational approach to oncology was all the rage, right? That's how crizotinib got developed. That's how multiple oncogenes, driver mutations got identified. I think we are coming to a point based on both the translational work, the scientific work that's being done, as well as basic molecular understanding of autoimmune disease, that people are segmenting the population. People are understanding what the driver cells, for example, are. And that forms a very fundamental scientific understanding, which historically was not there. So that's why it's coming to a sort of a convergence of that scientific knowledge, ability to develop trials to prove out the hypotheses, clinical hypotheses that multiple of these companies have, and also a more defined understanding of clinical endpoints in multiple trial settings. So given those convergences, I think we are at a point where 20 years ago, for example, people would treat SLE as a sledgehammer. Not anymore, because of these companies. So maybe what we can do is let's have the panelists' opinions on, including the disease states that they are targeting, on what makes autoimmune disease more amenable to this quote-unquote personalized approach of therapy.
So maybe I can start off with kind of how we're doing it, how it relates to that. So we have a unique platform that focuses on targeting natural pathways in the liver to re-educate the immune system. So our lead program in celiac disease, we are re-educating the body to a natural, an epitope that's found in gliadin, the dominant epitope of gliadin. So we're using that as a test model to show that we can tolerize against that antigen, which is responsible for the immune response that you see when patients with celiac disease eat a gluten-containing diet. So this we're going to look at in the clinic, we're going to look at also symptoms, but also the immune response to a gluten challenge, which is easily measurable. There's a variety of T-cell stimulation tests that you can use. There's ELISpots, there's whole blood assays, etc. Pending that, we also have a follow-up program in MS, which is a little more complicated. MS, there's a variety of antigens that are implicated in driving the autoimmune response in that disease. We've taken a subset of proteins that we believe can be used to re-educate the immune system. Now, may not be the solution for all patients, but when we start doing our clinical trials, we'll learn more and to see what other additional antigens we want to pursue.
So one question, follow-up question there, right there, given the disease states that you mentioned. Celiac has a well-identified epitope, others don't. Yep. So what kind of translational work would you think would be important in at least identifying the driver epitopes in diseases that are not currently characterized? Is it even possible to do that? Or in the future, would we have disease states that are defined by epitopes and disease states that are not, and then the clinical development and the PD responses to interventions worked out differently between the two?
Well, those are good questions. I think the preclinical models are always challenging, particularly in the immune system. I'm sure many of my colleagues on the panel will agree. I think for celiac disease, there's a variety of models depending on which HLA you're targeting. There's DQ 2.5 and DQ 8, I think it is. But we've tried to use a variety of animal models, both in using surrogate molecules for mice and also non-human primates to show that at least the platform can work. Obviously, if you do that across a variety of animal models, it gives you confidence that when you test it in humans that you have a fairly good chance. I think you're absolutely right that in celiac disease, the offending protein or antigen is well defined with gliadin. And there's probably, we've used our technology to sort of hone in on a particular signature that we'll use to re-educate the immune system. For MS, there are ways of getting at that. We've taken samples from patients with MS to see if they have baseline responsiveness and react to specific peptides and use that as a rationale for moving forward. It may be that if we find out that those antigens are particularly important for a subset of patients, then you could lead to a diagnostic test that you would test patients prior to treatment with the agent, which would be basically personalized medicine, if you will, for patients with MS.
Right. So just to reiterate what John mentioned, I think this is a recurring theme that comes up in terms of being able to identify the epitope or the driver epitope, and the subsequent translational work leads up to identifying a subset of patients who would presumably be more responsive to the therapeutic intervention. So maybe moving on to you, Gwen. Cell therapy-based approach is completely orthogonal. I think one of the very few, if not the only, company that is going that route. So how did Cabaletta Bio land on that? What was the genesis? And what's the advantage of a cell therapy-based approach in re-educating the immune system vis-a-vis other traditional modalities?
Yeah, sure. So yes, we are the first company using effector T-cell therapy in autoimmune disease, so that seems a little orthogonal for sure. But it's based really on sound science off of the CD19 CAR technology, which was born in the University of Pennsylvania. The construct was developed by one of our scientific co-founders, Mike Malone, and as the data started coming out from those clinical studies, Amy Payne, our other scientific co-founder, who is an expert in B-cell mediated autoimmune disease, specifically pemphigus vulgaris, she asked Mike, hey, is there some way that we can apply this technology towards specifically targeting the B-cells that cause pemphigus vulgaris? And the ability to do that really came out of an understanding of the specific target of the disease. So pemphigus vulgaris is one of the best well-described B-cell mediated autoimmune disorders. It has antibodies that bind specifically to a single protein called desmoglein-3. And so the innovation in our technology is being able to take the CD19 CAR construct and the autologous manufacturing method, which is now approved by the FDA, and changing the external portion of that construct from an antibody fragment to actually just the auto antibody target. So we call these chimeric autoantibody receptors, or CAARs. And so we have now, completed our IND, and we're moving into the clinic. And we think that this has a potential for a curative potential based off of the findings in the oncology studies. But there's that's what we'll learn this year in clinical studies. So.
Wonderful. So that's when I tried to landscape the universe of different approaches, that turned out to be this cellular re-education, if you will, turned out to be quite unique. In terms of, as we think through, Gwen, the downsides of a cellular-based approach, how would you characterize that given the CAAR type approaches? What do you anticipate to be the challenges in defining, for example, the patient population that would benefit out of this?
Yeah, that's a great question. I mean, in our case, the patients are already very well defined. There's already a clinical assay that characterizes DSG3 antibodies in patients, so the patients are already pre-selected for us. But there's a lot of unknowns, clinical unknowns and risks associated with this therapy in patients that we are intending to test. First is, this will be the first time that we're actually infusing engineered T cells into patients that have pre-existing immunity against those T cells. So the safety profile and understanding what happens in patients is something that we've carefully designed the clinical study to evaluate. And also infusing patients with activated T cells, infusing patients with autoimmunity with activated T cells has a risk of disease flare as well. So these are things we're looking at. They're things that we do have mitigation strategies for, and I think that's the real sort of thing to look out for in the upcoming couple of years, is really how does this modality work in patients with autoimmune disease.
So lots of translational work and pharmacology work in sort of understanding the first patient responses after you go in.
Yeah, exactly. I mean, monitoring patients for cytokine release syndrome and really characterizing what that is, understanding if you do see some sort of skin toxicity or some sort of toxicity like that, whether there's the presence or contribution of the T cells. We have a series of translational studies planned out in order to understand if that's happening, and then, be able to, implement modifications to the product in order to, take the next shot, or we might get lucky. So that's the test.
Perfect. So Bing, moving on to you. So Bing was a part of the MedImmune inflammatory group. He led research and translational there, and then spun out Viela. And Viela has since then been essentially a rocket ship, raising capital and not only proving to the public investors that there is a deep pipeline there, and including the first, I think, success from the NMO trials. So maybe, Bing, please describe a little bit of your pipeline, which is, I think, very broad and deep, and then what's the underlying overall sort of overarching principle in that pipeline?
Yeah, absolutely. Viela Bio was founded about 2 years ago. Essentially, it's a continuation from the work we have been doing at AstraZeneca MedImmune. I was the head of inflammation autoimmune for 7.5 years at AstraZeneca MedImmune. We have developed really a deep pipeline in the autoimmune area. Vela Bio currently has 5 molecules in development, 3 of those in the clinical stage. The lead product, lead molecule is targeting CD19, broadly B cells, and particularly plasmablast cells that are making autoantibodies. So that's a cell type. We have took that one actually quite innovative. We actually did not do early studies in AMO, but we felt the mechanism fits really well, high unmet medical need, and did a pivotal study with the AMO, neuromyelitis optica, a severe neuroinflammatory disease affecting the central nervous system. Had a positive outcome and filed BLA and is waiting for approval in the US. We have a PDUFA date of June. And now we have a molecule that's really highly active and is really consistent with our pathway-centric approach. Now we know that this molecule works well, blocking that particular pathway. We are expanding into multiple other indications. We are studying 3 other indications for this lead molecule. Behind that, we have a molecule targeting a different pathway, a T-cell, B-cell interaction co-stimulatory pathway called VIB4920. Had a paper published in the Science Translational, including the phase 1 data in rheumatoid arthritis, but that's not the indication. The lead indication we are now into is Sjogren's disease. It's currently in phase 2B study, also in a transplantation study. The third molecule is targeting an innate cytokine pathway, targeting PDC, plasmacytoid dendritic cells, depleted that cell type. That cell type is known to be playing a pathogenic role in a number of different types of autoimmune conditions, including, for example, lupus, Sjogren's disease, lupus nephritis, scleroderma, others. We have been creative, even in our phase 1 clinical study. Rather than go one indication at a time, we are going for this shared pathway. We have 5 different types of autoimmune patients recruited even in our phase 1 clinical study, phase 1A and phase 1B. So those are the 3 clinical molecules. We have 2 other ones currently in preclinical stage.
So one theme
rich pipeline.
Yeah, indeed. So one theme that emerges from Bing's description there is, the pathway-centric approach and the commonalities between the underlying pathology, cellular pathologies of, say, Sjogren's versus lupus and so on. So how do you think that the basic understanding of the pathology has evolved over the last 5-7 years, and how will it continue to evolve that will enable the approach that you are taking as it goes into the clinic?
Yeah. I spent probably over last 20 years or so in the autoimmune disease area. It really has evolved. You used to be looking at lupus. It's a syndrome. It's not even a disease. Highly heterogeneous patient population. So then begin to, a lot of work done by collaborations between academic institutions, companies, now begin to understand what is that pathway, the activation leads to different types or subtypes of that condition. I think that's making great progress. One really good example, as I mentioned, is the interferon, type 1 interferon gene signature. So you can use a signature to subsets, just as, for example, 70% of patients have this signature. What about other 30%? So that actually is a breakthrough. I think that was, multiple academic groups identified the gene signature and use that to subtype the lupus. So now you become more and less heterogeneous. So then you develop therapies targeting that pathway. Then you ask the question, what other autoimmune conditions also have activation in that pathway? So you go beyond the clinical phenotype, you are now going for molecular phenotype. That's really what we're trying to do, is go for molecular phenotype and release the treatment to the pathway.
Perfect. So that's another very important emerging theme, I think, generally in the space, that you go for molecular phenotypes as opposed to historically defined clinical phenotypes. Moving to Ruben, for full disclosure, Ruben is one of the, or Polyneuron is one of the NEA portfolio companies. We invested in them, I think, earlier this year. No, in 2019, I'm sorry. Can't keep my years straight. And what was most interesting to us was this, people take cellular or pathway-based approaches, but in some ways, what Ruben was doing was like in vivo plasmapheresis. So I'll let Ruben describe the approach, but it was completely orthogonal, and yet somehow the proof of concept was sort of there with the Rituxan treatment of this disease state, which has increased IgMs floating around in the circulatory system. So Ruben, perhaps please describe Polyneuron's approach, and then we'll go into why it's orthogonal and how you're going about developing the product.
So I'll try to explain our therapeutic approach. So we work with a platform which we call antibody catch, and behind this very simple name are polymers that carry antigen mimics and that are able to selectively mop up and pull autoantibodies into a degradation pathway in the immune system. So you can, with such polymers, you can selectively deplete a population of antibodies or a monoclonal antibody against a specific antigen, and all of this in vivo. Yeah, and apparently this approach convinced NEA. So we work, for you to understand, we mainly focus on neuroinflammatory conditions. So we work in the field of CIDP, GBS, anti-MAG, multifocal motor neuropathy. So neuroinflammatory conditions affecting the peripheral nervous system. That's where our core clinical expertise is. We're now moving with the first program towards the clinic fast. We have an IND pretty much ready for filing. This is in anti-MAG neuropathy, and as Bibas mentioned, these patients have IgM antibodies against a specific antigen. We designed a polymer with an antigen mimic, and as Bibas mentioned, there is clinical evidence that responder patients have a relative reduction in the pathogenic autoantibodies beyond 50%. The non-responders usually do not show any change in the antibody titers, and that was really kind of supportive data which we use now to move towards the clinic. Yeah.
So let's step back there for a second, just pause on Ruben's answer there for a second and think about it. So you have heard between the four panelists, there were four very distinct disease states that affect very different tissue types, from peripheral neurological tissues to the GI system. So the manifestation of the autoimmune phenotypes were in different tissues. And then that's point number one. The second point, which was also very fascinating to me, is that the commonality among them was, if you knew the epitope, driver epitope, there was a certain pathway, and there was a certain translational approach. And if you didn't know that, there was a different translational approach. I think on the other end of the spectrum, the traditional approach, if you have seen, for example, Aurenia's result in lupus nephritis, that was probably one of the very few successful ones over the last several years. But all these companies are taking approaches on the other end of the spectrum. And finally, the common theme that emerges is patient stratification. Either, as Gwen described, something that's inherent into the system, in which there are circulatory approaches, or for that matter, Ruben's approach for anti-MAG neuropathy. So based on these sort of common themes, and at the same time the diversity of different tissues and phenotypes in which we are seeing the disease manifest itself, one question that comes to mind is, from a regulatory perspective, all of you will probably end up going to different branches of the FDA. So how well, right. So how does, beyond this molecular understanding, which all of you are aspiring for and hoping that the external environment clinical environment will lead to by the time your products get into the clinic, how do you anticipate on the regulatory framework being supportive of this very fundamental approach and different approach from historical approaches that companies have taken in terms of the clinical need and the translation between the molecular approach and the clinical need addressed by various diseases? Maybe John, you can start.
Yeah. Assuming Gwen probably is facing the same group that we are. OTAT, are you going through OTAT? Yeah, same here. We filed our IND at the late part of last year. Now, I don't know if that pertains to my fellow panelists here on the far end, but I think that when you have an innovative therapy, they sort of put you into that group. When you have a molecule that's not a clear, small molecule or a standard biologic molecule, we're sort of a hybrid because we're sort of a complex molecule that includes a sort of a polymer-like signature which is attached to our antigen that we're trying to tolerize against. So I think the agency, when it comes to new sort of very innovative approaches, I think they throw in that catch-all. I think the approach from the agency depends on you know, I think it's our job in the industry to educate them about new technologies and to explain the science behind it, you know, and of course, as we move into the clinic with these innovative therapies, to assure that we are monitoring the safety of patients and have safeguards in place to assure that we, you know, are on top of any sort of non-fortuitous or, you know, challenges that may happen in patients as they go about treatment. Does that help?
Absolutely.
Maybe you want to answer, because we have I mean, we're going about it sort of similarly. You're bringing these exogenous, you know, T cells. We're trying to educate endogenous T cells to not react to the antigen. So, you know, we hope to induce, restore natural immunity, therefore, to hopefully get to the underlying cause of disease, to, you know, which is the goal that we want to cure patients, right? And you're doing it with exogenous T cells.
Yeah, so we definitely have the benefit of working with the FDA, and they sort of have reflected back to us their learnings in cell therapy. So I'm pleased that my sense of the FDA, you know, working with OTAT, is that they have learned learned what works in terms of efficient clinical development for cell therapy, and then especially rare disease. And I think we're benefiting from that learning. We had, you know, I think in our case, we're working with a rare disease where there's not a lot of expertise at the FDA, and so there's an element of us not just needing to educate the FDA about our therapy, but also educating them about the natural history of this disease, which has been changing over time, and actually that increased knowledge is one of the things that has enabled our ability to develop this therapy in this setting. So I think and the other component is what we've seen with the FDA is more openness for advanced review. And so there's now multiple processes. You know, there's fast track, there's breakthrough, there's RMAT, and they all increase interactions with the FDA. And actually with cell therapy, which is expensive, slow, and also in rare diseases, not making a mistake in development is critical. So having those early interactions is key. And so I've been working in engineered T cell therapy for almost close to 20 years now, and I've seen a real sea change in how the FDA is approaching these therapies. So, yeah.
Sure, yes. Given the number of diversity of the diseases we're targeting, we have been interacting with many different you know, divisions of the FDA. Our lead product is in the neuro division, and others are in the respiratory autoimmune area. And another division we're interacting because some of the autoimmune disease are in the derm division too. I have to say that it's really a great collaboration. And now, back to 5 years ago when we were doing the AMO study, so there was no approved therapies. There was no control studies done. There's no definition of the, for example, you want to measure the reduction of the attack. There was no definition. We worked with the agency on the definition. We worked with the agency on the trial design that gave you know, input on what kind of design. Do you need a placebo-controlled study, add-on study? So that was really a collaboration you know, with the agency doing that. I see the same now with the other divisions too. We have been interacting with them recently on one of the indications. There was no approved therapies. There was no clinical endpoints, but the FDA gave a very detailed feedback on what they like to see. It's really more collaborative now.
Excellent. And I know Ruben hasn't been in the quote-unquote lab yet. We haven't really interacted with the FDA yet, but speculating, given the complexity of the product that Ruben is developing, sort of analogous to John in some ways, how do you anticipate what would be the sort of the key highlights that you would work with FDA on? I presume it will be same along the lines that Bing described, very collaborative and iterative.
Yes, I think key points in this. So we have been interacting with the EMA, not with the FDA so far. This is due now, more than due. So highlights will be also educating the regulators about the disease. It's an orphan neurological disease that hasn't been on the radar of the agency, and educating them on key biomarkers in the disease. I mean, all of our programs are really targeting specific autoantibodies that are causally associated with autoimmune diseases. So the biomarkers are absolutely key, and generally in the field, I think the search for biomarkers is heavily the efforts are heavily increasing, and for polyneuron this is key. It's all biomarker based, and we hope to see more flexibility from the regulators in the future with regard to approvals based on strong biomarkers, and then potentially having the ability to bring more data post-approval. But in the best case, I mean, you hit the clinical endpoints and the biomarkers that you, yeah.
Excellent.
Biomarkers and educating about the disease.
Right. So once again, you see a common theme emerging there, I think. So now that we have done a couple of rounds here to hopefully give the audience a sense for the trends going on, why the disease states that individual companies are tackling are amenable to the type of modalities that each of these companies are going for. Maybe before we sort of view into the future and ask for sort of more speculative views from the panelists, maybe open it out for a quick round of questions if you guys have one. Yep.
No mic.
Well,
partly a commentary on the. Thank you. Partly a commentary on this meeting. It's been great timing. Could have been sooner, but I represent Imupharma. We've been in lupus, and we're in phase 3. We found out the hard way a year and a half ago, and it was very much a disappointment at the time, but when one looks at all the clinical data, you start to see why perhaps it didn't work. And so the pathway approach is very critical for us, because the mechanism of action is fundamental to many other autoimmune disease states apart from lupus. So we're actually in the process of looking at that. So I would be highly supportive of that approach. And also, I guess one of the questions would be, given that this is similar to, if you think about oncology 10 years ago, the development of newer approaches, immunotherapies, and combination therapies, what are your views on potential combination therapies in the future? Thank you.
Yeah, I think that's really consistent with you mentioned also the pathway. Once you understand, one way is to segment. You have one disease, you have these different pathways, you segment them into each pathway, you develop individual inhibitors for each one of those. But often you have, same patient, you could have multiple pathways active, so that would require a combination therapy. I think the people in this field are a little bit slow in terms of embracing that. Part of it is because early days when you have TNF, IL-1 combination that result in a side effect. But if you look actually what we're doing now in trial, most of those are in fact combination therapy. You have small molecules at top of that biologics, you are doing that. The question would be if you do, for example, cell therapy or biologics plus biologics, would that be? We actually begin to explore that. In one of our phase 2A study, we are combine the two of them together. to see whether you could increase the efficacy and also look at the safety profile too. I think that's one area for the future. But the key here is understand what are the pathways driving that disease so that you can combine.
Maybe just, I mean, as an oncologist, I mean, when we do, we're big combination people, if you will. But I think the success in combinations in oncology has been driven when there's, when it's not empiric and when it's rational based on mechanisms. So I think as we learn more about the pathways and mechanisms of specific agents, then one could lead to very rational combinations of treatment for various autoimmune diseases. For example, there are some of us that are working more at B-cell components of the disease versus the T-cell components. And so you could potentially envision that in a disease where it has both, you may combine two approaches.
And I would just add on top of that, so definitely for diseases that have a B and a T-cell component, you're going to need combination, but also even just for cell therapy in the pure B-cell mediated autoimmune disease, like one unknown for us is the role of soluble antibody, and so we might need to use combination therapy with even some of the emerging therapies in this area in order to actually provide that potentially curative cell therapy. And then there's something a little bit more unique for cell therapy, which is editing the T cells so that they can actually be more potent or be more safe as well, which you might consider a combination therapy as well.
Hi, I have two questions. I would like to have comments from the panelists. Two emerging area for autoimmune and inflammatory diseases are targeting inflammasomes and microbiomes. So I know there's some startups working on that. What do you think are challenges working in these two emerging areas to tackle autoimmune and inflammatory diseases?
I can take a first stab. Just to sort of set the tone. So great questions, thank you. In terms of microbiome-based approaches, the general theme that I have observed after having reviewed, primarily, let's say, microbiome in the gut affecting, for example, action at a distance in the brain or the periphery, understanding or precisely mapping, or having the ability, rather, to map the effect of changing the local ecosystem either locally or having its effect distally becomes very critical. So in other words, there has to be a good sense for what change does the intervention bring to the local ecosystem and the local effects in inflammation at the gut interface, for example, or at the distal place. And that cause-effect relationship in case of microbiome is not always mapped out with respect to the effector molecule that is being changed. Now, there are a few approaches where that effector molecule is well characterized. And in those instances, understanding the pharmacology of the intervention becomes much more detailed and evident. My personal view is if you don't understand that the effect of the intervention and the pharmacology, the effect, particularly if you go into the clinic too soon without understanding that, it becomes too much of a nebulous effect. And you can't really go back to map out what went wrong or how would you set the dose. Fundamental questions like that become difficult. So as long as there is an approach, and please send me one if you find one, that maps out that, that type of sort of a linear relationship, if you will, it becomes tractable. To your second question, which was around inflammasome. In similar ways, yes. Please go ahead.
Maybe I can, when I was at Novartis, I worked on Alaris, which is an IL-1 beta inhibitor. Probably for CAPS, it was approved and worked on the global approval for SOJA, which is systemic juvenile idiopathic arthritis, as well as the inflammatory syndromes such as FMS, HIDS, TRAPS. And I think that it's a great target because in those patients who have that particular perturbations of their inflammasome control, getting and squashing the inflammatory response and when they have an infection or an offending agent is very important. There are other companies other than biological approaches that are looking at the cytokines that are associated with that, but they're going at some of the core pathways. I think it's NAPF, NFL, PA, 3. I mean, Novartis has been continuing to work on that from a cardiovascular standpoint because of, and same with gout. But it's a good target, I think, and we'll understand the biology of it. I don't think we know the understanding of why people have that. We're getting at the genetics of it, that maybe the molecular defects that patients have that make their inflammasome respond in an aberrant way.
So just to augment that, in terms of the spectrum, I'm sure you're familiar with the NLRP approaches, various NLRP approaches. Gaspardin is also coming along, and there is, what I haven't seen, and perhaps someone out there is doing this, is a systematic analysis of the different NLRPs, the different ligand target mapping to those, and what effects it's bring. Because otherwise it becomes difficult to sort of, once again, zoom in and understand the precise pharmacology. The other aspect of this is, which is very interesting, and there has been, major transactions in the space with, the IFMs set of companies. I think there are 3 now, which I think Novartis, 3 different pharma companies, including Novartis, have purchased, and Atlas Ventures being the beneficiary of all of them, having seen the vision and built out those companies. It's a very interesting space, but ultimately I think it boils down to which patients and which disease states and in those specific patients do you target. The other target that comes to mind is P2Y7. And this was, I think, initially targeted in RA. It failed. It was tried to repurpose it into ulcerative colitis target because of its presence in the interface in the gut, but once again, it's a difficult target pharmacologically. I don't know if others have other insights to either microbiome or inflammasomes. Okay. Hope that helps you.
Chemistry challenges.
Yes. Yes, please.
Hi, thank you. Just a quick question. How available or otherwise, and how much of limitation is natural history data sets in these indications, which I know also sounds a bit counterintuitive because that's something that's always really important in rare disease drug development, and yet what you guys are trying to do is you're kind of taking that concept into much more complicated multifactorial diseases. Is it easy to get data sets that are well validated, big enough in size so that you can kind of track the effect of particular antigens or particular proteins in these diseases?
Yeah, thanks for that question. Yeah, no, that's a great question. In these rare diseases and also with a very rapidly changing treatment landscape, it's very difficult to have a comparator data set. And in terms of, what will your clinical development look like? How fast do you go? How do you do a control group, especially with cell therapy, is a huge challenge. So there are no contemporary databases that we can draw from that I think that at least my sense of it is that we could have confidence, that the regulators would have confidence in. And so this is a very active area of discussion for us. How will we do that? And I also think in the face of a rapidly changing landscape for standard of care with these patients, that's an additional challenge as well.
Yeah, I would say disease homogeneity and available natural history, at least to a certain extent, was key in selecting our programs. It's absolutely key. Yeah.
How do you manage the narrative of autoimmune diseases versus oncology? Because autoimmunity really was ahead of immuno-oncology for immune modulation. If you look at the 80s and 90s with thymoglobulin and all those different tolerance-based therapies, having developed at Core Pharma the nanoparticle platform there and struggled with trying to get through to big pharma's depths of marketing people to appreciate that maybe there is a good reimbursement to be made. How do you approach that, especially with the cell therapy in a space where chronic treatments are the norm, and you're essentially going against a wave of entrenched dogma within this space?
Well, I actually think that for cell therapy, I mean, that's actually one of the attractive aspects of autoimmunity for cell therapy that makes cell therapy feasible, is the fact that there still are not curative therapies for these diseases. And so patients have to have either recurring or chronic treatment, and that is costly. And a lot of the new drugs that are coming out are quite costly, even though they're effective. And so if you actually stand up the cost of cell therapy with chronic treatment with some of these therapies, I'm not sure I want to call any of them out specifically, but it actually makes cell therapy attractive, but I think for any indication for cell therapy, it's attractive if it has the curative potential or a long-term remission potential. And so that's what we are hoping for based off of the data in oncology with CD19 CAR, which is against B-cell mediated cancers, to use it against B-cell mediated autoimmune disease and try to affect the same type of long-term responses.
I think if you look at the current therapies for autoimmune disease area, it's chronic therapy. Yes, it is chronic therapy. A lot of progress being made. If you look back, when the TNF was approved for and expanded to other indications, I think one of the areas that a lot of companies are looking now, including ourselves, is just can you induce some kind of tolerance for the future, do an induction and or maintenance kind of a regimen. That's really back into the combination therapy. Can you use combination therapy to do that? Another area could be, can you treat the disease early? One of the pathways that we are looking at CD40-like and the CD40 interactions. Granted, it's a preclinical data. When we did the preclinical data, if you treat the animals early in life, before the onset of the disease or early onset of disease, actually you can just give them a couple of doses, then they're cured. So that's one area is combination therapies, etc., to do a tolerance. Then you don't have to treat them for life.
So, Bing and Gwen gave the logical answers. Can I give a cheeky answer? So I mean, this is partially true, and I say this in jest, but what you're seeing here among the panelists is people who have purposefully taken orthogonal approaches. If there was a need for 286th immuno-oncology target, these people are not your people. So that's first of all. This is on purpose, that they are going after from a very first principles-based approach to medical needs. And second, before you talk to the pharma marketing people who are boxed in on the traditional TNFs and the small molecule chronic therapy approaches, many of these won't necessarily fit that paradigm, right? And that change eventually will come to the people who will be marketing these products, presumably pharma. Maybe the smaller companies will do them themselves. But if you start with the pharma marketing people who are boxed in with a certain paradigm of treatment, will perhaps never get there, and many innovative therapies won't get there. So that's my soapbox on there. But these are innovative companies, and they change the paradigm of treatment based on a fundamental science and unmet need, and will come to modifying the pharma-based approach that we have had historically.
Maybe just to open up and make sure it's not such a cloudy day from a big pharma. So I'd say I personally find that there's a lot of interest by big pharma in our company in particular. We have a transformative collaboration with, we initiated a collaboration with Celgene, which is now BMS, and there's still high interest of that. I think big pharma is maybe trying to get a diversified portfolio as opposed to oncology, and I think we wouldn't be here if we didn't feel that there was still a lot of unmet need in the autoimmune space, and I think the innovative approaches that we're all working on, we hopefully will have a big impact on those diseases. The one thing about autoimmune disease, they've been around for a long time. except for maybe pemphigus. I mean, pemphigus has been out there, but it's still not been addressed. There are a variety of PROs and various instruments that you can actually see if you're having an impact on the disease, and I think that gives us an advantage where PROs are not really readily available or have been used for approvals with agencies in the past. So I think it's, I don't think we have to have the narrative versus oncology personally. I think that we can focus on the things that we're trying to do to help patients.
Completely agreed. Yeah. So,
just very quickly to also put the umbrella away. I mean, I think some of the pharma companies have made investments for cell therapy specifically, and so it actually, I think, is something that they are more likely to go towards in order to leverage the investment they've made.
So given that we have 5 more minutes, maybe we can do a quick round on what's coming. What's the shortcoming right now, biggest shortcoming in your respective disease states or approaches, scientific approaches, and what's coming down the pipe that would put more spotlight in the approaches and the disease states. And I say that from the perspective of, maybe you can put it in context, like for example, immune tolerance network, the concept of central and peripheral tolerance has been there forever. In Ruben's instance, the concept of plasmapheresis and taking out certain disease causative or at least correlative antibodies also has been there forever. People have had IVIG businesses for a long, long time. And those are all sledgehammer approaches. IGIV till today is made from plasma sourced from humans. And 3 companies run billion-dollar businesses out of those, hospital products that are made out of those. So if Ruben is able to change that, let's say, what would be the critical aspects that you would see coming in the next 5, 7, 10 years that would put increasingly more spotlight that is beginning to be shown on autoimmune disease states? Start with Ruben.
I think it will be very hard to wipe out IVIG from the market, particularly as we are taking such a focused, directed approach towards these indications we go after, to the platform being basically limited to monoepitopic diseases. We can obviously not compete with the IVIG or these more general approaches in any conditions, but I think we will move there step by step, and I'm convinced the market will change in the next 20 years significantly, with some of these more general approaches falling away for very targeted approaches, such as the ones these companies here are developing.
Yeah, I think the challenge for autoimmune disease, one is, again, we often do treat it as one condition as one, and you have to do large trials, which is making it harder to recruit the patients. The second one is each of those autoimmune diseases, unlike oncology have this pretty much the same endpoints, right? Survival, PFS, etc. Autoimmune, each condition, you have different endpoints. Lupus is different from Sjogren's from different from others. So that's the challenge. You have to have the larger studies. So then you have to analyze. So that makes your chance of success may not be that high. So my prediction, what we are doing that now is go for subsets of in each of those. So that way you have a defined endpoints, you have a shorter trials too, smaller population. So get the drug to the market faster. So I would predict that more drugs will be approved for targeted patient population for the future.
Yeah, so I would predict that we're going to start seeing a lot more uptake of cell therapies with both investigators and regulators into the field of autoimmunity, applying learnings from oncology into these autoimmune indications, and I think we're going to know, certainly in the next couple of years, whether this effector T-cell therapy, what their challenges are and how quickly it might move. I also just want to echo, it's a very different way, but echo what Bing just said in terms of the FDA continuing to be proactive in understanding how to rapidly advance clinical development in this area, I think is something we're going to hear more and more about. So I think there's really a lot to watch in the next few years in autoimmunity. I think we're going to see sort of an explosion in this. We are seeing it. We're going to continue seeing it. in the next few years will be very exciting, and especially for patients who don't have treatments.
Just 2 points, because I know we're running out of time. Number one is biomarkers, being able to identify the patients who benefit from our therapies. I think a lot of the scientific tools and instruments to assess T cells, B cells, and actually get down to actually cells, specific cells, will enable us to identify the patients who are likely to respond to our therapies. And number two is improvement in patient recruitment. I think we can't do what we do and learn about the impact of our drugs unless we can get patients to go on trials, and I think they would see the benefits and possible disease-changing therapies that we could bring to offer, but we need them on our trials.
Fabulous. Well, thank you, everyone. Hopefully that was useful to all of you, and thanks for attending.